Wow, that was incredible. Both Dr. Richardson and Dr. Moore did fantastic talks. And I know we have a harpist coming up, so we want to make sure we get the harpist on time. But there are several questions, and it's great to see Drs. Richardson and Dr. Moore. I know, it's great to see you guys. All right, I'm going to start with Debbie Richardson. So there are a couple questions that, I mean, and if you guys, because we can't see the participants, but if you guys have questions, please send them in. We've gotten a few PARP inhibitor questions, not too many that I can tell so far for EDCs or for immunotherapy. But Debbie, I'm just, I think with all these new drugs finally being approved for our patients with GYN cancers, especially on the antibody drug conjugates, because the ADCs that we're using in GYN cancers, mirvetuximab, tisotumab, et cetera, are different than other antibody drug conjugates that are being used in breast cancer or say lung cancer. And how are you at University of Oklahoma kind of educating your staff about the differences here and making sure that we're not lumping all of the antibody drug conjugates together?
Thanks for that question, Ursula. So that was a great question. Basically, the companies themselves have been fairly helpful as far as asking who needs to be educated. We're also somewhat lucky, Katie and I, that we have a PharmD embedded into our clinic. So we did do some education, both from our PharmD, from us, and utilize the company's help to educate them about especially the eye care. Dr. Moore, my partner here, had done the original phase 1. In fact, she treated the first patient in the world on mirvetuximab. And so we had a lot of experience using that drug already in our phase 1, but then now we're just moving it into our late phase. And so when tisotumab got approved, the eye care wasn't that different from what mirvetuximab, other than using those the eye packs and one additional eye drop regimen, that vasoconstrictor. So our PharmD actually put together the 3 eye drop things, and we just send our patients down to our pharmacy in our cancer center, and they pick up the pack. And in fact, and then we educate the patients as well. And of course, the first patient I treated forgot her eye drops at home, and so we send her back down to the pharmacy because you can't treat them without those things. So it does take a little bit of preparation, but once you get treat a couple patients down, it gets much easier for everybody. But there's a learning curve, I think, for our staff, whoever supports people in the office, and then also in infusion.
Yeah, that's great. And I think we were really fortunate to have the accelerated approval of mirvetuximab. I think that things happen pretty fast, but that companion diagnostic, I know, is back up and running, and several labs have access to doing the companion diagnostic. So for our patients, they should be able to access mirvetuximab now. Would you agree?
I do. I ran into that problem. I ordered several, they got done, and then all of a sudden there was a shortage. And so, but the company again was quite helpful in telling us which labs had reagent and moving the tissue around, for people that it was more urgent.
Right.
And so I pretty much have results on just about everybody I've ordered it on this point.
Yeah, it sounds great. Good. And then Debbie, a question from the audience from Colleen Bridge. She asks, what is the duration of NED allowed to enroll in the UpNext study?
Thanks for that question. So this is meant to be a maintenance after chemotherapy. So in other words, somebody is platinum sensitive, has gotten a platinum doublet with carbo, for example, and something else, and then is just finishing that chemotherapy. And so within 4 to 12 weeks of finishing that platinum regimen, and somebody has to get at least 4 cycles of platinum in order to be eligible, and then also has to be NaPi2b high. So it's not somebody who's been prolonged NED. These are patients who have recurred and have been on treatment, and then it's maintenance right after finishing the chemo, essentially.
Gotcha. And then another question just came in about, would you guys both be able to provide a copy of your slides?
Mine is at Let Every Woman Know.
Okay, so but you're okay giving that out. Okay.
I think it should be fine. I think everything I presented is in the public domain.
Yeah, so we maybe can go back and just check in, but certainly something will be available to the Alaskan group. Okay. Dr. Moore, great talk as always, incredibly insightful. A number of questions have come in on PARP inhibitors. So the one question is the PARP inhibitor information that you presented just for epithelial ovarian cancer? And have PARP inhibitors been tried for other types of ovarian cancer? So I guess I'm almost thinking, well, you can answer that question, but I think then another question came in on sort of low grade versus high grade cancers.
Yeah, those are great questions. It sort of just brings up, can you hear me okay?
Yes, perfectly.
Okay, so to make sure I have
Yeah.
new headphones. It brings up just sort of the difference between sort of how we do clinical trials and then what you do with that information once medicines are available sort of more broadly. So the clinical trials for PARP inhibitors as maintenance therapy were all done only in epithelial ovarian cancer, and then more specifically only in high-grade serous. And some of them allowed high-grade endometrioid as well. So these are all what we term TP53 mutated tumors. So TP53 is a tumor suppressor gene that is lost in every high-grade serous. It's just, if your tumor doesn't have loss of that, it probably isn't high-grade serous. So it's just one of the things that identifies it. And so that classification identifies patients or with tumors that may be vulnerable to PARP inhibitors. So the main indication by trials is in high-grade serous and high-grade endometrioid. If you look at the labels, it just says advanced epithelial ovarian cancer. And so that led to a number of practitioners saying, well, I can use it in clear cell, or I could use it in mucinous, or I could use it in low grade or grade 1-2 endometrioid. And I think what I would say about that is that we have zero data for use in those settings. And the reason the trials were limited to high-grade serous predominantly is because that is a specific type of ovarian cancer that has an inherent vulnerability to how it fixes its DNA. And when you give a PARP inhibitor to it, that's synergistic. Clear cell doesn't have that in general. Low grade serous doesn't have that in general. And so it's not that we're trying to like deny access to these medicines to other tumors. It's just that they're not likely to work, in my opinion. And so I'm not saying it's wrong if it was tried, because we all try things sometimes that aren't on label, but there is a rationale for why it's limited.
Yeah, makes sense. Good answer. Okay, so for upfront PARP inhibitor maintenance use, and this is a question that comes up, what about patients with stage 1 and stage 2? All the trials that you've mentioned and or run have been in patients with advanced stage 3 or 4 disease. So what about for patients with stage 1 and 2?
So this is a great question. And I get it all the time. I'm just going to start off by saying we have no idea. There's no data.
No data.
And there never will be, because it's just, we have as a organization run clinical trials for patients who have early stage ovarian cancer, stage 1 or stage 2, and we lumped them all together: serous, mucinous, endometrioid, which makes no sense now that we understand cancer better. But even by lumping everyone together, those studies took a decade to do. And so it's just not feasible. That being said, have I done it? Yes, I have, with extensive counseling. There is some retrospective data, meaning at sites that see a lot of women who are at high risk for ovarian cancer because they have a BRCA mutation or a RAD51C germline mutation, or some others. We're learning more about these, and they have undergone risk-reducing surgeries or surgeries and have these grade 1 tumors. There have been these studies where they say, okay, I have 100 of these. I'm going to look at how they did. And so this has been done mainly at the University of Washington with our colleague, Dr. Elizabeth Swisher. And what she's reported, and this has been published, is that women who have low-grade serous ovarian cancer that is serous and BRCA have a much higher risk, it seems, of recurrence than a stage 1 serous low-grade or low-stage cancer that's not BRCA. And that kind of makes sense, because if a woman has a germline mutation, it's not just in the fallopian tube, it's kind of everywhere. And so could it be in the peritoneum? Could it be other places? Makes sense. And so the risk of recurrence looks to be about double based on this retrospective study, which is low validity data. It's good study, but it's, it's not prospective. But based on that, I do talk to patients that have stage 1 ovarian cancer who have a germline BRCA mutation about use of PARP inhibitors. And I tell them all the risks and benefits, specifically that there is a low but present risk of secondary malignancies like leukemia. And so I could give you a drug that maybe you don't need, and I could cause a cancer that will kill you in 1 to 2%. So it's a very careful counseling discussion, but a lot of my patients want it. For a non-BRCA low stage, I would not use it. That's stage 1, fully staged, appropriately staged. If you're stage 1 because you had an ovary out and no one went back, hell yeah, I'm going to use it because you're likely not stage 1. And stage 2 is advanced, and I would definitely use it there, even though we don't have the data. I would use it, and I don't lose any sleep at night. But I'd be interested in what you do, Dr. Maciulonis, because I respect your opinion a lot here.
Yes, yeah,
and we may not have to do the same thing.
Yeah, no, I think, I have a couple of patients who are on stage 2. And I think, as you so, well talked about in your presentation, that PARP inhibitor history kind of data is coming out, and we've had FDA changes and new issues have come up. So I think I am probably not using them on stage 1 patients now. So stage 2 is a discussion, and you're right. Like, is the patient completely staged, et cetera, and then analysis of the risks and benefits. And this comes to another question. I know we have a couple other questions coming up, and I want to make sure we get that we don't go over too much more time. But so for the upfront patients, the niraparib data, PRIMA, used 3 years of PARP inhibitor, 3 years of niraparib. All the olaparib studies have used 2 years of olaparib. And I know, so an extra year of PARP inhibitor from year 2 to 3, obviously excess toxicities, impact on quality of life, and also financial toxicities. How are you kind of rectifying or sort of kind of bringing together those two data sets when we know that the 2 PARP inhibitors are equivalent in terms of efficacy? If someone starts an niraparib, are you going to go over 3 years? Or are you going to say, you know what, the olaparib data said 2 years, let's stick with 2 years?
Okay, I'm going to try and answer this complicated question quickly because I see Dr. Hardesty.
I know, I guess
she's going to pull me off the stage.
The harp isn't right.
Melissa, can I answer? Do I have time? You may stop.
Yes, I want to hear what you have to say about it, actually.
Okay. But this is a very difficult question because there's really, when we selected these links of therapy for the clinical trials, it was arbitrary. I'm just going to tell you that we had to, the FDA said you can't do it forever. And so pick a cut point. And we did have some science behind it, but it really was like, let's try 2 years. And then PRIMA said, we're going to do 3 years. And so know that there's no data behind it. And what I'm going to tell you is that at this meeting, I'm at our national NCI ovarian cancer meeting. I'm at the airport flying home, and there was a concept presented for women with BRCA. Right now it has HRD in it, but for women with BRCA, going a trial of 1 versus 2 years. So not more, but less. And why do we think that that's interesting? It's interesting because if you look at the survival curves that I showed you, I know briefly in the Solo 1 presentation, the curves become parallel at about a year in BRCA. The event rates, how often is someone recurring? It's about equivalent at a year, which suggests to us that perhaps the benefit, if you're going to cure someone, which is what I want to do, if I'm going to cure somebody in frontline ovarian cancer, it's an early effect. I've done a surgery, I've gotten it all out, I've done my chemo, NED, and there's some cells floating around, and I knock them out in the first few months of PARP and then continue it for however long you make up this year. But if I've cured you, I've cured you at that point. If I haven't cured you, which 55% of patients on Solo, we didn't cure, and so we have to remember that and fight for them. does it make a difference to do 2 years or 3 years? Or am I just exposing you to extra things that are going to make potentially your response to subsequent lines of therapy lower? We don't know the answers to this. So I go to 2,
Yeah.
and I don't go to 3 unless I have evidence that someone has disease. Like if their CA-125 is kind of high, or they have peritoneal thickening and I know it's there, but it's stable and my patient's feeling good. I'm treating disease there though.
Right.
I'm not in maintenance. I'm treating cancer. I'm going to treat it until it starts to grow, and then I'm going to go to something else. Those are different concepts, but I really think less is more here, which sounds like blasphemy coming from me, but I stop at 2.
Yeah, I agree with you. I for patients who are on niraparib, they get to that second year. I have a very frank conversation with them, and a couple of people have said, "Look, I want to go to 3," but I think most people have said, "I want to just stop at 2," and I agree, given all of the olaparib data.
Yeah, I think if a patient's so nervous and they want to go to 3, I'm not going to lecture, but I don't do an indefinite Parp. I do not do that.
It's so hard to be the hook here because
oh, sorry.
I just thank you guys so much for everything. And now I'm the hook.
Thanks for having me.
Yeah, thanks for having me too.
Yes, Dr. Richardson and I both ran long in our videos. We're sorry.
All right. Thank you guys 100 times over, and we look forward to seeing you at the next meeting. And
take care.
We're going to move on to the next section. Thank you, everybody.
All right.
Bye,
Safe
everyone.
travels, Katie. Bye.
Thank you. Bye.